good morning. Dada, good morning. Sir, good morning. Nirita, good morning.
Good morning. I was keeping the camera and the speaker off to get a better bandwidth.
Yes.
Good morning to everybody. Yeah.
Good morning.
Yes. Our principal sir is over here. I request Dr. Gautam Kundu to kindly grace the event and give the welcome note and please introduce our notable speakers for today. Thank you, sir.
Thank you, Shatabdi. Right at the beginning, I'm a bit sorry because it got a bit late because of the connection. So I'm really elated to say that the Physiology Department of Vidyasagar College, in collaboration with IQAC, has been able to organize this physiological webinar, which is of a national status. And this particular webinar, the credit goes to the head and the convener, Dr. Subhadeep Ganguly, and also to other members of the department, such as the IQAC coordinator, Dr. Alok Ghosh Chaudhary, Dr. Chandana Sarkar, Dr. Oendrila Chattopadhyay, Dr. Sanatan Misra, Dr. Monalisa De, and Ms. Shatabdi Ghosh. For this particular webinar, I welcome all the guests who have come today to hear two eminent speakers, which we have. And the topic of today's webinar is higher brain function. Well, when I was a bit young, one of my teachers had asked me one day that can you name an organ which grows 10 times its original size? Well, I was not able to say that. But later I came to learn that it was the brain. The brain generally grows as we grow. And the topic is very interesting today, the higher brain functions. And the first speaker, eminent speaker who is here today is Professor Birendranath Malik, who is a retired professor of neurobiology, School of Life Science in JNU, New Delhi. Presently, he is also working as a professor and director in Amity Institute of Neuropsychology and Neuroscience, Amity University. And Professor Birendranath Malik actually is a very well-known person in the field of physiology because his career speaks volumes about him. He has never secured second position in any of his exams. So he's a gold medalist in both in his BSc and MSc studies. He also possesses a diploma in homeopathy. That's a very interesting thing that at the same time he has got a diploma in homeopathy. I think he must be having a lot of persons who often come to him for some medicines. And another interesting thing about him is that in his school days, he was a good athlete and he used to play cricket, hockey, and was a member of the scout team also. He has established neurobiology research lab and has initiated REM sleep, which happens to be his topic today, REM sleep research in our country. He has published about 140 research papers and book chapters. He has actually written 4 books during his tenure. He has received various awards, including the prestigious Shanti Swarup Bhattacharya Award, which he received in 2001. Well, it gives me great satisfaction, and I'm really honored to have him today among us. and to hear from him about the REM sleep or his particular field of work. Our second Professor Tushar Kanti Ghosh is also well known among us because he's a visiting professor in the Department of Physiology, University College of Science and Technology, Calcutta University. And he also has been working on brain and immunity for the last few years. He has published a number of research papers also and has actually guided a number of students during his illustrious career. And today, his topic, Alzheimer, cognitive impairment, Alzheimer disease. This particular Alzheimer is a progressive disease, as we all know, that destroys memory and other important mental functions. So, particularly the brain cells degenerates and dies. Whereas, contrasting to this particular topic, our first speaker topic is evolutionary significance of REM sleep. Well, REM sleep increases the functioning of the particular brain. So today's topic, which we have higher brain function for the motion and against the motion. So for the motion, Professor Bhirendranath Malik will be speaking, and against the motion, it happens to be the against the motion because of this Alzheimer, which is a lower brain function, in fact. With Professor Tushar Nath Malik will be speaking on it. Now, coming to the particular REM, it was a bit new to me also. REM means rapid eye movement sleep, or it is also known as REMS. It is a unique phase of sleep, not only in mammals, but also it is seen in case of birds also. Now, it is related to rapid random movement of the eyes, which is accompanied by low muscle tone throughout the body, a state known as REM atomia. It leads to sleep to dream vividly. Now, REM phase of sleep is also known as paradoxical sleep and sometimes as desynchronized sleep. REM sleep is physiologically different from other states of sleep, which are known as non-REM sleep. Now, REM and this particular non-REM sleep alternates within a particular sleep cycle, which lasts for about 90 minutes in adult human. Now, regarding this topic, I'm sorry that I won't be able to continue because I saw other works, but I have certain questions over here. Does REM sleep have any influence on a particular person who walks in his sleep? Because I have seen this happen in one of my relatives also. While we were all sitting, he was sleeping, and suddenly he got up and started walking towards the door
Sir, will you please repeat the question once more?
to sleep. Does this brain has any connection with this sort of walking in the sleep, or for persons who go on talking in his sleep? Often we do talk, and when we get up, we hear that you are talking about this particular thing. So this talking in sleep, is it also connected with REM sleep? And another thing is the activity. Certain activities which we perform in the daytime gets carried over in our dream. And often it happens that the body starts functioning accordingly. For example, a football player often starts kicking someone who is besides him and then shouts goal. So this particular functions, are they related with REM sleep? Well, I think it will be a very interesting topic, and I welcome Professor Birendranath Malik as well as Professor Tushar Kanti Ghosh, who will be the second speaker. So I hope this particular webinar will obviously will be a very grand success. And let us start with our Shatabdi. So over to Shatabdi. Please continue. Thank you.
Thank you, sir. Thank you for the kind words. Now I request our IQAC coordinator, Dr. Alok Ghosh Chaudhary, to kindly say a few words.
Thank you, Shatabdi, and good morning, everyone. Today, we are fortunate enough that Professor Birendranath Mollik and Professor Tushar Kanti Ghosh are among us. Professor Mollik is a great name in the field of modern REM sleep research, and his original scientific contribution in this regard has been acclaimed throughout the world. On the other hand, Professor Ghosh is one of the best teachers I have so far seen. Being a student of physiology, I am really fond of listening to their lectures. I believe that we will enrich ourselves today. And many regards to both of you, sir. Apart from this, I must mention here that even in the harsh time of current pandemic situation, the Department of Physiology of our college has arranged its second consecutive webinar. Thanks are really due to all of our departmental colleagues, especially Dr. Subhadeep Ganguly, the head of the department. And now I invite Shatabdi to take care of the next part of the inaugural program. Over to Shatabdi.
Thank you, sir. Okay, now we proceed to the first technical session. I again inform all the delegates to write their queries in the chat box, and our eminent speakers will answer accordingly. So as both our eminent speakers have already been introduced by our principal, sir, but very briefly, I would like to reintroduce Dr. Birendranath Mollik once again. So Professor Birendranath Mollik is Professor and Director of Amity Institute of Neuropsychology and Neurosciences, Amity University campus in UP. He retired as professor and was the Dean of School of Life Sciences, JNU in New Delhi. His research mainly focuses on understanding neural regulation and functions of REM sleep. He is renowned nationally and internationally in the field of REM sleep. Professor Mollik is a proud recipient of several national recognitions, including Shanti Swarup Bhatnagar Award, Medical Sciences, CSIR in 2001, National Bioscience Award, DBT 1999, Shakuntala Amir Chand Award, ICMR 1992, BK Anand Award, APPI 1984, and JC Bose National Fellowship, DST 2010 to 2020. He served as invited faculty, guest faculty in several universities in India and abroad. Mainly, to name a few, several amongst the universities in abroad like UCLA in USA, Harvard University, USA, University Autonoma Madrid in Spain, Wrocław University in Poland, including Marcel Decker, USA 1999, and the Cambridge University Press, UK 2011. During his school days, he was conferred with the President's Scout Badge, also represented district and divisional school teams for selection to the state teams in hockey and cricket. He has proposed the evolutionary significance of REM sleep. Based on these findings, he has hypothesized that REM sleep has evolved in higher animal species to maintain the noradrenaline at low level in the brain, an antioxidant compromised organ, and protects the brain from oxidative overload. So Professor Birendranath Mollik will talk on evolutionary significance of REM sleep. On behalf of our department, I gracefully welcome Dr. Professor Birendranath Mollik to deliver his speech today. Welcome, sir.
Okay, thank you very much to everybody. This is in a unique era that we are living. Probably at our own space, we are around, but speaking to n number of people from different places, different locations. And now I guess since I cannot see all of you, can you please give me a feedback that you are able to hear me? Can you hear me? Can you see me? Then only I will know that things are
Yes, sir.
Yes, sir. You are audible as well as visible.
Okay. Thank you very much. Okay, I am quite pleased to be here this morning. And now so many names have been said. It is by nature, I cannot recollect so many names, but thanks to the principal, Dr. Kundu, and but Dr. Shubhadeep, who has been in touch with me, I think, for several months. And then all the organizers and the IQAC chair, Dr. Ghosh Chaudhary, and rest all the speakers. I welcome all of you and I am pleased to be here. Now, I have a little problem here. So many things have been told about me. I think now I have to do the justice. And now let's see how much I can do about that. And I will be glad to take your questions. So it is now about 10:25. So next about 45 to 50 minutes, I'll be talking about why I am saying in my proposed hypothesis. And then certainly we will take on from there as and when. Second thing is, in between, if there is any interruption and if somebody has any burning question, please do interrupt me, just for clarification if required. With this small introduction and respect to all of you, I will start my talk. Here, I'll now present the slide. Once I am doing it, please let me know that whether you people can see that. All right. Now, can you see the slide now?
Yes.
Yes, sir.
Can you see it is moving?
Yes.
And I'm proceeding with this now. Okay, as I said, it is a unique era that we are living, and particularly last a year or little more, that wherever we are and we can talk, though we cannot see each other directly in person. So today I will be talking about evolutionary significance of REM sleep. What does it mean? It is not that I am saying how REM sleep has evolved, but then what I will be trying, which has come to me of late, maybe say 5-7 years. Why did nature create REM sleep for us? So I will be presenting that today. Now, while doing it, let us see. Many of us have seen this type of phenomenon. Now, here if you see, is it diseased or it is normal or if it is what? And if I say that it is somebody sleeping, you may or may not agree. But one thing we can understand that here these phenomena, certainly the muscle tone is lost. And therefore, this is happening. In addition to that, you will find here that there are eye movements, and the children, even in the dog, that is in higher mammals that we can understand. Now, these were many of us had seen. Now, if I say this is in the sleep, within sleep, this is a picture I am showing, a picture from, it has been published in 2010 from Frontiers. These are the different animals. One can understand they are sleeping, but then how do you confirm they are sleeping? For example, if I tell that, okay, go and touch this tiger, I don't think anybody would dare just like doing it, because you don't know whether somebody is awake or anesthetized or dead or whatever. That means the major problem was there was no objective criteria to analyze them. That was because nobody knew if the brain has electrical activity. Once that was identified, that was one was able to record. One recorded, for example, here is the cat. This is record from my lab, but not that I am the inventor. So you have EEG. Let me bring the pointer. Yeah. So you will have EEG, EOG, EMG, that was electroencephalogram. I am sure physiology students would know all these. Electrooculogram, myogram, and pontogenicular occipital waves, but at this moment we can leave that aside. Based on these electrophysiological signals, one could identify what I showed, that the heads are drooping. Yes, indeed, EMG, the third channel, is totally flat. Yes, there are atonia, therefore probably that is happening. And this happens in humans, cats, and I'm showing you even in the next one, which is in the rat. This is in the rat, you see EMG, the third channel is totally this. You could record it even in the animals. This is in my lab, so you can see the head stooped. You can see here the atonia is coming. This is EEG, and here there are eye movements. In addition, there will be these eye movements, what you saw a few minutes back, the child as well as in dogs. See, there were significant eye movements, my point. Now, when was it known? This was known, it was 1951. This is the gentleman where Eugene Asadinsky, who first described this. And the great name in this field, another second great name was Michael Jouvet in France. None of them is alive at this moment, but they discovered it. Once this was there, you can see these eye movements and eye movements in the dogs as well. Because of the eye movement, it was named as rapid eye movement sleep because it was within sleep. So this was discovered in 51. And as you would know, that it takes at least 50, 20, 25, 30 years to come to textbook at knowing any of these subjects for general mass. So we were passing out in 74, 75. There was hardly anything known about this. That was one luckiest part for me and why I picked up this topic. Now, my lab focus was how does brain regulate REM sleep? I'm not going to talk about this now because so many things is the volume. If there are 25, 30 PhD students, average 5 years, there will be about 150 man years worth. So, but I will relate them and that will have a significant connotation. I can speak about that when the principal raised some issues, though we do not know everything, but we might have some answer to it, which I will talk about at that time. The second thing I was interested in, functions of REM sleep. I didn't have any iota of thought process when I started in mid-80s, the evolution. But later on, when I talked about function, and it is not that I am the only person talking about function. There are many people who talked about like REM sleep helps in memory, REM sleep helps in brain. Like if the great Nobel laureate Crick had talked about, it clears the unwanted memory, unwanted something which goes whole day in our brain. Then some people talk about thermoregulation. Some people talk about brain development. So based on my function, because my studies, I was thinking why it is such an important function, which is a basic for our living system. It cannot be, we cannot say only one or two things that's function or 10 different functions. It has to be an instinct function. So I will be talking about that. In order to go about it, let's see here. How did the world evolve and the life evolve? Here, if we talk about from the sea and the first cells and going into evolution, the human is coming at the end here. And there we are talking about some consciousness, which we talk about when relatively higher. We don't talk about so-called quote unquote within the word of consciousness in an amoeba or something, though they have some type of phenomenon. That way, even some physicists may say consciousness of the cosmos word. I'm not talking about that. We are talking about in the biological sense. Here, this consciousness broadly, we talk about wake and sleep. And wake and sleep, these are the very basic function which is there in all living cells, even all living systems, not only cells, from single cell to the highest animals, so-called humans. Now, if that is there, within this, when people started talking about recording physiologically, I talked to you about the electrophysiological signals. Then in addition to wake and sleep, one could find REM sleep. Now, there is the that is the inflection point, what we have to understand my hypothesis. Now, if we talk about this, see, and I'm giving you some example. Oh, this is not moving. Where is my this? This video is not running. Let me see. Okay. I'm sorry. This video suddenly is not working. It worked. Okay. What I am saying, all of us have seen sometime the ants remain quiet and then sometimes they move. Now, we might say they are sleeping or they are taking rest. This video was like that, which unfortunately something happened that is not working right now. It worked even an hour back when I tested my all slides. In any case, here is the fish. That means what I am saying, yes, you will find sometimes this fish here, see, it is stopping. And after a while, it will start moving. It is in mobile. Can you see this? It is written in mobile. Then it starts moving. So what I'm saying, sleep and waking or rest and activity, as it is mentioned here, basic rest and activity cycle is a fundamental of living process. Now, when this was going on, when REM sleep was discovered, whichever higher species it has been recorded so far, be it cat, dog, elephant, even birds, it is there. That means it is one of the fundamental things, episodes, what is happening for the living system. Now, somebody may ask, can we identify REM sleep in that ant? See, REM sleep as an experimental physiologists or scientists across the globe, we have to define certain things so that you can show it to a third party outside. Now, since it has been defined with EEG, who has a reasonable brain, that is a technical limitation that we cannot record EEG. But there I will tell you, we have recorded even slight mode of EEG, even in zebrafish. Recently, we are doing in the lab. Now, if it is there, whether we can do it, the REM sleep also, because REM sleep has been defined by EEG, EOG, EMG. Now, there is a difference, which I will not deal into at length, because there is some other possibility that there is some acclimatization, whether you can have REM sleep to that level when those animals who are not sleeping in a protected environment, so that they are not killed by the predator. That is another way of understanding, which I will not deal with. Today, I will deal with what is fundamental, why REM sleep has come evolutionary. Now, this REM sleep, as I said, it is there in all species so far recorded. Now, this REM sleep has evolved about a million years ago when it diverged to mammals. It is in more in babies and reduced with age. All of us know this. One of the very interesting thing is, you name any disorder, it's simple fever to complex disease, REM sleep is affected. Of course, somewhere it is reduced, somewhere it is increased. We can talk about that later as mechanism, which I am not talking at this moment. Second thing is, REM sleep is regulated by neurons located in the core of the brainstem. Look here, when we talk about any function by the brain, the paleocortex, paleo-brain, is responsible for most fundamental things, including feeding, including rage, anger, sex, all these behaviors. And heart regulation, respiration regulation, all is there at the center of the brain and the brainstem, which is then modulated by the higher brain function. So we have to understand that. Therefore, it is, I would think evolutionarily it has been there. Now, why it should be there? And it is affecting, as we can see, this increased, if we deprive experimental REM sleep or for some disorder, many of these like irritability, aggressiveness, confusion, anxiety, these are increasing, and memory function, maturation, all these are decreasing. So what do we understand by it? One of the thing is here, it is the excitability. If they are all by the brain, then the brain neurons are different than any other somatic tissues. It has an excitable property. So what even then, in 90s, when I started with, I had hypothesized within myself. I didn't dare, because that time I didn't have the expertise and experience. I couldn't talk about this, but I have made this slide maybe in 2000 or so when I have the data. But it was in my mind that REM sleep cannot be of vestigial significance. But then why has it evolved? If it is there, which function, if it does not have any fundamental function, why it has not been lost in evolution, and that too in species, not only in the same species. So why has it evolved? Which fundamental function it would serve? So before I started, people only knew about REM sleep loss alters different functions. Then I talked about, if at all it is function, my lab focus is, I talked about this function is one of them. And then question is why and how. So what I thought, if it is a fundamental function, it is affecting the neuronal excitability. But the excitability can be in the short term or long term. Means if I have not slept even for a few hours or 1 day, 2 days, I am irritated. There's something I might do any mistake. And that is one of the reasons you get road accident and otherwise. But the point is, if I am supposed to have a long-term implication, like your thought process or your memory processes or development of the brain, it has to be at the structural basis. So that is what is the global implication. How would it happen? So I will not talk about all those what we have studied, but I will show you at least a glimpse, and then we will go for some part at depth. So my hypothesis was REM sleep maintains brain excitability, and excitability, one of the things what you maintain is sodium potassium ATPase, and we showed that sodium potassium ATPase is increasing. Now, why should it increase? If sodium potassium ATPase, that is an enzyme, and it is in the membrane, and if it has to be affected by REM sleep is a behavior, even if it is getting affected by the brain or modulated by the brain. That means what I hypothesized, that REM sleep disturbance would affect basic REM sleep regulatory mechanism. And if regulatory mechanism, then that would do certain things, which in turn will be affecting at least in the brain. And then the whole body might be affected with some either neurohormone, neurotransmitter or hormones, or it will affect other blood pressure. Something can go and it will affect the whole body system. Then I needed to know the basic REM sleep regulatory mechanism. We worked, as I said, we did work on that. And then we had shown that there are neurons which are REM on and REM off. Many of you have been knowing or might have heard my lecture sometime or must be studying these in your master's level. The REM off neurons, this is, of course, this slide is from my lab, but I am not the inventor. That was shown in 1974. These neurons stop firing during REM sleep. Unlike any other, these are locus coeruleus neurons, noradrenergic, and during REM sleep deprivation, they keep on activity. So if they are keeping on activity, then if neuronal activity means neurotransmitter release, and if the neuron had to stop, the neurotransmitter would not have been released. So neurotransmitter will be reduced. But since deprivation, this neuron will keep on firing, neurotransmitter will be excess release. Therefore, my contention was, if anything related with REM sleep loss or REM sleep functioning, then at least it could be many other things which we did not know, nor we know all right, even now. At least one thing will be that noradrenaline level will be changing. That was my idea even in late 80s, early 90s when I started. Based on that, we worked out several, at least 6, 7 theses. And then we worked out this is the model when noradrenaline works and then ATPase activity. This is how, and then we worked out the intracellular mechanism. We have seen at the gene regulation level, which I'm not talking about. Interestingly, noradrenaline affects gene mRNA stability. Not even mRNA is increasing. That would be a whole lot of stories, which I'm not going to talk about. But I'm just giving a glimpse of, because many people might be interested, or to stimulate many people that you can do many things like that. And that is having stability by our one, these types of uh, transcription factor modulation, which we will talk about in some other time. Now, as I said, that is for temporary behavior or changes. Now, if it is for long term, like next speaker will be talking about Alzheimer's, we talk about any psychiatric disorders, quite often, unless it is depression associated or REM sleep behavior disorder, there is REM sleep disturbance and increase or decrease. But there are many instances like Alzheimer's, Parkinson's, there are degeneration of the neurons. We do not know the mechanism as yet, but my study will throw light that why it should be. Here it is, there must be some changes in hardwiring. Now, quite often these are in humans where you can only see post-mortem. Where is the technical, as an experimentalist, I have to show it what is happening in any system. And we do it in the animals with all due respect to the animal life for human benefit. So this is what we wanted. But then when I started in late 90s, well, I did not have the facility. So what do? What I did after deprivation, we did simple microscopy, simple histology. We did first paraffin sectioning and then went for nasal stain sectioning. By the way, when we did nasal sectioning in late 90s, over the years we developed now nasal sectioning, nasal stain, well, it's not nasal stain, we are talking about Golgi stain, but that I will talk later, sorry. So we did this simple nasal stain and we, with a simple microscope, we took the pictures. And this is for those people, forgive me, those senior people here. I'm talking about those who are starting. Quite often people talk about, oh, I do not have this facility or that facility. That paper says this to do 2-photon microscopy or something, 2.5 crores. Well, you can do that if you're lucky, but then you have to see something which is very difficult to do and to interpret, and there will be a lot of false data. So you have to be expertise. I'm not against that. But if you have in a simpler system, it is easy to understand until you become more or gain more expertise. But here, one of them, we took the section, and as you can see here, this is locus coeruleus where noradrenergic neurons are there. But here we did not stain the neurons with antibodies, so we do not know whether there are noradrenergic or otherwise. But this was the first, since it's the publication 2005, this was studied maybe 2003, 4, sometime like that. And then look, LDT PPT area, medial preoptic area, lateral septum as a control. And here you can see free moving, and then actually REM sleep deprivation, this positive control, locus LPC, large platform control, recovery, and then prazosin. Remember, I said, in order to understand the mechanism, and if it is related to REM sleep, in my understanding, there has to be effect due to increased noradrenaline. So if I am giving an alpha-1 blocker, we did with beta blocker as well, but that did not affect much. So here, this actually is changing in the sense that will be shown here. This neuron, one of the neurons, you can see the size has increased as compared to this control. It is not the same neuron. Don't ask me that whether it is the same neuron, because you have killed the animal, sacrificed the animal after deprivation. So I will no way be able to say that. But we can do it in a culture, which I will show you later towards the end of the structure. Mind it is in early 2000, and this is about mid 2110 or something, where it took 20 years or 25 years for me to develop that and the facility. And we did that. It did increase and then it is coming back. But in prazosin, it is tending to come back. Now, this is one neuron. You will see here, these are 1,000+ neurons in each of these we have done. And painstakingly, my student Sudipta had done those with this type of microscope. No automation. But we can see here, as you will find, the dotted line here is deprivation with this triangle. Here are, these are each of the average, the area of those neurons. And then it is increasing. Here the histogram of about 1,000 neurons in each of these areas, and that is happening. Then we try to do, well, I might have removed it. Then what we have done antibody stain with a TH hydroxylase, tyrosine hydroxylase, to show indeed here these are noradrenergic neurons in locus coeruleus, as you can see here. Cholinergic chat staining in this LDT PPT, it does decrease. So it is not just a mistake, because if it was a random process, then you wouldn't have got this type of change, and it couldn't have been somewhere it is increasing, somewhere it is decreasing. Those are significance we can talk later, but it does happen. But that is not the main, why I'm saying we'll talk later, because this is not the main focus. Main focus is how does it affect, so that neuronal structure will change and how we can protect that. That is a significance. We did even then 2006 paper, confocal microscopy with double leveling. Yes, the structure is changing. So, and then that suffices with this electron microscopic that there is blebbing, that means it is going into apoptosis, but we did not know the apoptosis then. Mind it is late 90s and early 2000. Later on, we did with antibodies and all, we will show later. Here, this is with the, that was 2006, and here it is 2016, with again electron microscopy to show how the mitochondrial cristae is getting damaged. If it is apoptosis, that is getting damaged. And you can see here the nucleus, how there are chromatin condensation and chromatolysis is taking place. But the good part is it cannot be 5 hours deprivation and all of us will go this, but there is a process of recovery if it is within that limit. I will talk about that more there. Then if it is there, how do we go about that, what we say like traffic accidents, our concentration losing and every other thing? Look here, we have done electron microscopically CA1 areas. More you can see here. We have talked about synaptic cleft, the size here. We did electron microscopic, and painfully we have estimated for about, you can see here, about 80 synapses, average 80, 85 synapses from multiple brains of those, and these are control and others. You can see the length of active zone is increasing. Means length of the synapse structurally may not change, because once the synapse is formed, it is not that it will change just like that, because it is developmental. But what is synaptic? What is active zone? Active zone is the length where there are presynaptic proteins and postsynaptic proteins, where you can see about MUNC18 and postsynaptic density PSD 95. Here, indeed, after deprivation, these proteins are increasing. Means what we have talked about in electron microscopic here, that means the length, there is accumulation of more proteins. That means, to my understanding, as you can see here, average 229 micrometer, a nanometer, to going about 280 nanometer. To my mind, it is an adaptability or acclimatization the brain is trying to adjust so that the loss can be reduced and you are trying to recover. Otherwise, probably even after 2 hours or 5 hours deprivation, we would have gone lost. But then if we are continuing over the days, over the months, and then probably we are losing many things or some other means. And I showed it here, it is with prazosin, as you can see here, it is reducing. That means it is because of increased noradrenaline, which they are trying to adjust. All right, this is one thing, please remember. And how about the memory part? Because I have to show with functional thing. All of us knew that after REM sleep loss, there is loss of memory, but not technically, experimentally shown in that way. Even if it was shown, as you can see here, this will be the rat here. Normal rat, it is going to the platform after learning within a few seconds. Here on the right, after a deprived animal is going around and round and around. This was done even in some form by other people. I'm not the first person, but maybe first person in actually doing with day-wise deprivation. But more importantly, we took out those rat brain. Then we traced them three-dimensionally, which you can see in animated one. A neuron in CA1 neurons, we have done CA1 and CA3 both. See the branching of, or here it is CA3 neurons. I'm sorry, I thought it is CA1. CA1 also we have done. You can see we are showing in all around how much branching of a neurons. This is about 250 micrometer thick section. And then we traced. It is a painstaking job. It takes 8 to 10 hours or 8 and 9 hours to trace one neuron. And then see after deprivation in the right, how they are, these are randomly picked neurons. If you ask that whether it is the same neuron, no, I cannot. I don't think anybody would be able to do. But then I have then correlated with even with primary culture of neurons, which I will show you later. Now, these we have published just, I think, this year or last year and something. Some papers have gone, others we are yet to publish. So their branching is reduced. That is what you can see. In the control, it is less. And here, the lower panel, you can see the actual histology with the Golgi stain. Here when I was telling you about the Golgi stain, we have now done minor modifications, and that way the problems of Golgi stain taking about 3 to 4 weeks, we have done it in 24 hours. And then there were a lot of false positive, be it with glia and others, that is significantly reduced just by manipulating a specific temperature and whether perfusion is done before or after, or with perfusion, without perfusion. So that is, I think, 2012 or 13 paper that is worldwide being used, and it's very fast and one can do that. Okay, now this is the paper that is 2021. What I said, how different areas are getting affected. This is in CA somewhere, Soma area. This is what we are talking about. Well, now I have to, I guess you people understand, but I have to tell you what we are saying is that, let me see. No, I might have removed that slide. Okay, this is what is the soma that we are picking up. Then we are measuring the area. The lower side is the upper side, that will be the apical dendrite, and then branching of the dendrites. And these are the basal dendrites. That is what you will find here. All these areas, soma area, soma perimeter, at apical dendrite length, apical dendrite nodes number means where from they are branching, and then spine density. As a physiologist and others, you would know spine density is talking about spine, which are talking about the memory. Memory does not necessarily mean only that I remember your name or otherwise. It is for every function that we are doing. The neurons have to talk to each other for communication. That is the spine density. And then this is at the basal length dendrite. And you can see they are significantly reduced after deprivation. As you can see, the black one here, be it apical length, be it basal dendrite length, be it nodes, apical dendrite nodes number, and the spine numbers. Similarly, in the next one, where it is CA3 area, that you will find here what is happening. These things are reduced. So this is the first study. And then we also did specifically, the spines are also at least 3 types: the stubby spine, the thin spine, and mushroom spine. Mushroom spines are the most matured one, which talks about permanent memory. Thin spine, which is intermediary, and stubby when it is starting. And you can see here, thin spines are maximally affected. That at the process of memory formation and then going into memory, the permanent memory. So these are the first study we have to yet publish this. These are the first study that is happening with REM sleep loss. And these are the data which you will find. There are 4 sets of each experiments, where each of the neurons not less than 20, at least with 4 sets, means average 5 neurons from every set from each of these areas that we have studied. This is CA1. Similarly, for CA3 we have done. And these, all the red ones are getting affected. I showed you dendrites, the number and all. In the interest of time, let us proceed. But that means they are actually getting broken down, or the cells are damaged. Now, why should the cell get broken down or damaged? Then the functions. What we have talked about, the mechanism, as I said, we do not have time to talk about, but based on my studies, we have we have talked about and modeled even by mathematical formula and computer modeling based on the data, not the simple computer modeling from my thought process. It's based on the data completely, and whatever we are seeing, more than 80% data from my lab and part of my personal studies, where it was shown these noradrenergic neurons are continuously active during, you can see here, wake. When we go to REM sleep, activity is reduced, but when we go to REM sleep, that is noradrenergic neurons is totally shut off. And this is there, as you can see in the Progress Neurobiology 2012. But what I showed you, this comes what I have told you earlier, that the noradrenergic neurons shut here. Because of shutting firing, noradrenaline will be reduced. And if the noradrenergic neurons continue firing during deprivation, noradrenaline will be increased in the brain. But at that time, you may remember this was around mid to 90s, a hypothesis. But it needs to be shown. Then you will find here, this is what after this work is going on parallelly. This is 2017. We actually showed the red data is after a red continuous line is deprivation, while dotted line is normal. The green one is GABA, you can ignore for the moment, but at least for red one, indeed it is increasing, be it any area in the brain, be it frontal lobe, cortex, dorsal raphe, PPT, and locus ceruleus, but hippocampus, it is going down. This you might remember. The hippocampus, it is going down. One thing in hippocampus, there is no direct projection of noradrenaline from their neurons. But we did show that, yes, it is increasing. But then for increasing, you need, I will just not digress, but to give you the flair of science, we did even epigenetic transcriptional modulation studies, and we did show that, yes, epigenetic changes is taking place for the tyrosine hydroxylase and dopamine beta hydroxylase level. From it is increased acetylation and decreased methylation. Therefore, it is showing that, yes, indeed, TH and DBH is increasing, which will show that, yes, it is increasing noradrenaline. And then we worked out that this noradrenaline, we did the entire pathway here, how it is increasing the apoptosis by doing the apoptotic pathway, how the damage is taking place. Possibly because of this, the neurons are getting vulnerable. To get total damage, it may be neurons will be totally lost, which is over the years, which might happen in a very chronic condition, because you don't get Alzheimer's, you don't get Parkinson's just in one day. Over the years and predisposition, and if it cannot even do recovery. But then this is one of the causes. But so far, what I am saying, that we worked out after deprivation, and that to increased noradrenaline in this pathway is increasing. And then it is indeed, it increases here, like see, bad APAF1, where all these increasing caspase 3, cytochrome C, caspase 9, PAKT pathway, phosphorylated AKT pathway, but caspase 8 did not change, suggesting that one of the pathways, that is intrinsic pathway versus extrinsic pathway, that is working. Now, even if it is there, that we did because you are depriving the animal and then taking out the brain and doing Western blot, and you are doing it, that was reasonable. But as a physiologist, I am not satisfied. I needed to show that. If it is with the noradrenergic neurons, what we did, in the rat brain, we are injecting TH, tyrosine hydroxylase, siRNA or shRNA. We did with both. Here I am showing you a control study. In the normal animal, if you are giving, this is siRNA, this picture and lower picture here, if you can see my pointers, I hope you can see this. Whether you do with siRNA, you do shRNA. In a normal scrambled shRNA or siRNA, see, TH positive neurons in locus ceruleus is still existing, whereas if you do it in the siRNA or shRNA, after 8 days of shRNA or after 24 hours of siRNA, they are significantly reduced. That means you can expect that there will be noradrenergic projection areas, there will be less noradrenaline. All right. Not only that, we have punched out the specific regions of the brain, this pointer, as you can see, and that one on the left side western blot, as you can see, indeed there is tyrosine hydroxylase enzyme is reduced. Now in this one, I was talking to you about cytochrome C. Look here, if you are doing with controlled with scrambled siRNA and depriving, there's still cytochrome C is increasing. But if you give siRNA after deprivation, actual siRNA, then it is decreasing. This is after 24 hours of siRNA. And if you do it with shRNA, that after 8 days, then we found the similar thing. And for tyrosine hydroxylase western blot, also you can see the similar thing is happening. That means this noradrenaline from these neurons, when they are increasing in the system, they are causing this apoptosis. Right? This is what our finding. So far, so good, and we were very happy and all. But within myself, I cannot tell that to my student that, okay, we are facing some problem. But then once you are getting more something, you get more question. That is the beauty of doing exploring areas of science or questions. This is 11:04. I have some time still, right? Okay. So,
Yes.
okay. So the question then comes, if this noradrenaline is harmful, and this is related to REM sleep, and it is existing for millions of years and generations and evolutionary different species, why has it been existing? So that is what the question. If noradrenaline is apoptotic, then you can see, I showed you the slide there, evolutionary, how the universe came up and the animal species. See, initially in the annelids and others in the lower species, the neurons are only network. There's no big brain. That means the blood supply has to be separate. These are open circulatory system in these. And then it becomes voluminous, where the blood supply has to be there. But in the process somewhere, I don't know evolutionary why, there is less of this. As compared to many other tissues, brain is antioxidant compromised organ. And here you can see here there's a lot of oxidative load in the brain because of this high lipid content, less availability of antioxidant, high metabolic activity. Why high metabolic activity? Human brain uses 20% of the oxygen consumed by the whole body, but constitutes only 2% of the body weight. But it may have a lot of cells, but at a localized area. So what can it happen? Because it struck me, then is REM sleep doing something there? Because it is primarily for the brain. It is not just rest. It is doing something in the brain. Additionally, it can do many things, but the nature must have done something at the fundamental level. Then what we found, though it is oxidating is happening, but in deprivation, be it lipid peroxidation or ROS, they are actually reducing. Be it you are doing in a vivo, that means we are doing in the animal after deprivation, or to simulate that, we are doing it in a synaptosome preparation from animals. Then adding noradrenaline there, actually it is lipid peroxidation is reducing and ROS generation is reducing, which is prevented by primarily prazosin, not beta antagonist propranolol. Then we did, because if we have to go to the molecular mechanism, it is better to go to the cell line, because it is so many complexities will be there in case of the whole animal in the vivo system. So gradually we started cell culture system and we went into neuro 2A, and I will show you in the primary culture. Here indeed, if we are giving H2O2 hydrogen peroxide, there is whole lot of ROS generation in the cell line, but if you give noradrenaline, then H2O2 is significantly reduced, and these are the actual picture that you can see with DAPI and PI stain, and then they are merged. Then the question is, what can it happen? We found there are a lot of iron in the brain. Of course, it is in the liver, but there are a lot of antioxidant as well in the liver. But otherwise, in the brain, almost comparable to liver, but it is lacking antioxidant. Of course, therefore, probably the glia has evolved. That is what is doing part of the job. And this iron, what we know from chemistry, it goes from ferrous to ferric through Fenton reaction and goes into this hydrogen, this nascent oxygen that will in turn go into damaging the cells. And this damaging need not necessarily be at the cell body, any metabolic process, and therefore the branchings can be broken because, excuse me, branching will be broken. Excuse me. So how do we do this? And then important thing, this iron imbalance is involved in many neurodegenerative disorders, and many of them are correlated with the, okay, that slide I have missed. Okay, that is gone. Many of these diseases are REM sleep disorder, or in many of these disorders, the REM sleep is significantly affected. But as I said, you will not find any disease where REM sleep is not affected. But then there is a correlation that iron is involved in this type of neurodegenerative disorder. We do not know why, but then one mechanism is iron is helping this Fenton reaction. All right, and then it is inducing this, but that we knew from chemistry. If tried to do that, indeed, if in presence of noradrenaline, the absorption of the ferroferricin is reducing, that means noradrenaline reduces the antioxidant property. And we knew from the old chemistry 60s studies, because of the cyclic ring, noradrenaline does have an antioxidant property. So this created more puzzle in my brain and what to do. Then what we wanted to do, on one hand, the noradrenaline is necessary. On the other hand, noradrenaline is damaging. Therefore, there may be a finer balance, and that is there in most of the molecules in the body. And what we know of is feedback and feed-forward reaction. But then how will it work? In order to show that, it is complex to do it only in behavior. So I have to do it in cells. And if you are doing in a cell, it is better to do primary cell line so that I am more akin, similar to the brain. Then we gradually developed cell culture and primary cell culture. And when we were developing, if I have to do in the rat, unnecessarily we are killing a rat mother, or maybe doing it and we are killing many pups. Essentially, then I tried to develop this from the chick embryo brain. It took me several years, but then all credit goes to all my students. Of course, actually, I did personally this one once I saw in principle things are working, and then I gave it to my student, and they did a great job. From this brain, we did actually primary culture of the neurons, where you can see this is what the neuron is growing in a live cell, and possibly like synapse is getting formed here. And if this is happening, then you have now the way to test how much noradrenaline is doing. It is helping progress, or in principle, okay, the science we are doing in the table, in principle, whether things are working. So first thing to develop this. Then what we are doing is, this is the same, okay, again, video is not working. Here you can see, here it comes there, because I wanted to study. Look here, I am a REM sleep physiologist. REM sleep noradrenaline is reducing. You go for deprivation, it is increasing. Again, when we talk about deprivation, this is an abnormal situation, one may think. But what is happening essentially? Somebody is awake. One is going into, if you can see my finger moving up, one is going through the day. When somebody is awake, that means those noradrenergic neurons are active. So noradrenaline level is increasing in the brain. When it is going up, For some reason, it is decreasing. Now, physiology cannot work by whims and fancies. It has to be feedback or feed-forward regulation. That is in our humoral system, that is our neural system. So some mechanism has to be there, which the mechanism, today I don't have time, and I think all of you will be getting exhausted listening again, the technical, of course we have worked biologically as well as mathematical modeling. I could take another 3 hours to talk about them. So this noradrenaline, so to simulate that in a system, these are, as it is written, you will find primary check embryo neuron and effect of noradrenaline different concentration. Means 0.1 micromolar, 1 micromolar, up to 100 micromolar we have tested. Look here, as compared to control, here with lower dose of noradrenaline, all of you, I guess you can see carefully, branchings are increasing here 0.1 and 1. With 50 micromolar, they are all damaged. These are time-dependent, time-controlled. I think whatever a few days, 3 days, 4 days, and we have gone up to 3 months we could record, my student could do. So essential part is lower dose of noradrenaline, the branching are profuse. Now you can think of what I showed here. If this is branching, means what? If you see here at this point, something is coming off here, something is coming off there, and here it is withdrawn. Because of this branching, so essentially later on, it will go and make synapse. Or if it is withdrawn, the synapse is withdrawn. Think of a situation. If it is withdrawn, a synapse was there that will break down. And if it is progressing, it is possibility of synapse forming or not. Then it will be there whether it is the right target or off target. All those questions will come later. So here now it is in primary culture, as you can see here, up to 10 micromolar, even at 1 micromolar here, the branching is increasing, 50 and 100. This is decreasing. This is just published in 2020, this year, or maybe last year. I forgot the year. Maybe this year. This is in Journal of Neuroscience Research. And this is, we know the noradrenaline has a scavenging property. Now, this noradrenaline, as you can see here, then what is happening? We, with the modern technology, because of after having these gene connotation or these gene annotation, and then we are talking about evolutionary concept and bioinformatics, what even we did? Tyrosine hydroxylase, which ultimately, and DBH, dopamine beta hydroxylase, which ultimately is going to give you noradrenaline. But even from the insecta, you can have the basic minimum for the amine structure that is evolutionary conserved. But it has molded itself with evolution, and you will find tyrosine hydroxylase is there. All these vertebrate, or even at, of course, birds and other vertebrates, of course, at some point you are getting that in mollusca or platyhelminthes, what it is. So I will not be able to comment there whether they have REM sleep, but at least from protochordate and all, we so far know that it is there. I have, based on many of these things, I have correlated in a philosophical concept when we talk about with the philosophers how REM sleep has, or the thought process, or the consciousness where we talk about, and the role of noradrenaline. But at this moment, I will not go into it. But evolutionary, you can think REM sleep, at least we have seen in these. But here we may not be seeing that in notochordates. Here, those are not there. And we might have, we need more depth data onto this. But these are in principle as a policy why that evolution has taken place. Now I am towards the end of my talk. Let me see what is happening. Look here, evolutionary, as I had talked to you, I will go back to my earlier slides. I will go back to the slide was I said here. Let us understand. Life evolved at some time. The brain was like a network, and there was an open circulatory system. For some reason, with evolution, when the brain became more compact, And the services started, and the brain had to do many other function and parallel processing. For some reason, antioxidant is less there. The glia already there, who is taking care of antioxidants. And because of, and iron is somewhere there and in loads of there. So it is a oxidate vulnerable organ. But it is taking so much of oxygen and doing all the purpose. I'm talking about oxidative animals. I'm not talking about those anaerobic organisms. There it might be different. When we are talking about the cognitive function, consciousness, where the brain has evolved in that way, the nature had to create something. What would be there? You have there evolutionary conserved those amines, which it molded from monoamines to even dopamine to noradrenaline, which is there in the brain. And it has the antioxidant property. And therefore, if it is there at a very low dose, it is helping, or even at a higher dose, as I showed, even after deprivation, it is preventing the oxidative stress because lipid peroxidation, ROAs generation is reduced. And if they are reduced, it is tending to protect the neurons from vulnerability. And if it is increasing, the same noradrenaline is increasing the cleft, synaptic cleft, by increasing the postsynaptic density proteins or monk proteins, which is making the synapses. If the synapse length is increasing, it will help to cope up with some damage, but it can be an acclimatization process. But in the process when noradrenaline is increasing, nothing can go if it is, nothing can work in that way if it is going for a long process. Therefore, they will reduce the induced REM sleep in the process. When REM sleep is there, here you can see this graph, which is in Frontiers in Molecular Neuroscience just a year or two back, what we have proposed. There, the first time I talked about evolutionary concept as a fundamental process of REM sleep, not just memory, not just growth of brain or something, body temperature regulation. It's a fundamental process. Here, see, during week, noradrenaline is increasing. That is a normal process. And the level is at a certain level. A break point is about to reach when REM sleep is coming. So noradrenaline level is going down and the neurons start protecting. Though certain level of noradrenaline is protective, but when the dose higher is going, all these processes are through the alpha-1 receptor. We have data, but we have yet to publish it, but then the student has left. I don't think we will be able to publish, but somebody can pick up. Then the sensitivity of the beta receptor with higher dose of noradrenaline. And those beta receptors induces different type of apoptotic or the neuronal damage. And that probably sets in. And therefore, in the system, it does noradrenaline reduction. How will it do? It induced the whole brain mechanism that noradrenaline neurons will stop firing. If it stops firing, then it does some of the things. In addition, they have done certain things where it will do eye movement or where it will do the muscle atonia. And if those are not happening, some disorder. So they as are byproducts. But fundamentally, the REM sleep has evolved like as it is shown here with something when it increases REM sleep deprivation too much, that goes as a break point, then it starts neurodegeneration. Otherwise, normally it goes down the level, which helps in neuroprotection. And it is written here, low dose protects while high dose causes damage about the noradrenaline. And I showed you the mechanism, how even intracellular mechanism, how will it cause the damage and apoptosis and degeneration. So possibly REM sleep has evolved to maintain noradrenaline level in the brain, which serves as a housekeeping function of the brain. And therefore, evaluation of REM sleep, which is a non-invasive method, may provide an insight about the health of the neurons and the brain. And there is enough examples of it. there is REM sleep is getting affected during you. I talked to you about any disorder. So if we have, like we have fingerprinting, we have iris measurement for identification of person, I think that we can develop this. But then, yes, we need time to develop. What will be the REM sleep? And every individual will have a different type of REM profile. And then how is it changing? Like we do with age, what is happening to glucose profile, or we are talking about profile about for cardiac function, and we correlate what is happening 2 years back, how was it, and now what is happening, which way it is going, and it will have a prognostic value. I would think this probably will help to generation, to human mankind for leading a good health. In addition, how the fundamental studies, which will probably help in curative, I have shown enough findings at least to prove in principle. And therefore, it's quite some time in 2012, I have written an editorial, never sleep on sleep and don't lose sleep on lost sleep. Why sleep when sleepy, lest you become sleepy? With this, I will finish my talk, but it's my present duty to acknowledge all my students and associates who have done great job. And this is certainly at a level it has reached that you can see from behavior to molecule how things are working. And I'm grateful to all my students. And these are the students yet to submit. Raghunandan Yadav has already submitted thesis, and he's waiting for his viva. Others, because of this last 2 years, almost corona, some delay, but within, I guess this year, 2 or 3 of them must submit. Only 1 or 2 will be left. And I had several postdocs, 3, 4. One of them is Rachana, who did many of these, like Nora Dinadin and others. Bindu, who did the apoptosis work. And I am grateful to all the Indian funding agencies. See, the taxpayers' money has been invested toward doing this job. I think I have done some justice, but then, yes, some animals' lives are gone. With all due acknowledgement and to all humility, yes, we have gone for that is for greater cause. And now the last, but not the least is, yes, all my work was done in JNU, which I superannuated after about 36 years. It's just a month back. And I was asked I have been persuaded by this MIT, because they have the neuroscience department. They have been telling me last 1 year, so I have just joined after superannuating there. They have a nucleus of neuroscience. Let me see what best I can do. So I'm grateful to all these taxpayers' money, all my students, to JNU in particular as a whole, and now in MIT, let's see what I can do. And lastly, thank you to all the listeners, and I hope I have been able to convey certain meaning and doing some justice. I appreciate and thank you very much. I hope I am within time.
Yes, sir. Thank you so much for enlightening us with your informative and instructive speech. Now I request you to take a few questions, 1 or 2 maybe, for time limitation.
Yes, please. Go ahead.
Anyone have any query? Please send your questions.
Can you hear me?
Yes, please, I can.
Okay, sir. I would like to get your opinion about the 2 things which are slightly different from today's discussion. The first question is, we usually dream only during REM sleep, not during non-REM sleep. Is there any underlying definite mechanism that's why we dream only during REM sleep? This is the first question. And second question is there any current literature regarding the relationship between gut microbiome and REM sleep? These are the 2 questions, sir. I seek your opinion.
Okay. As regards the first question is whether we dream during REM sleep or non-REM sleep as well. Earlier, we used to think that the dreams come only during REM sleep, but the concept is changed now. We do have dreams during non-REM sleep as well. But what we understand now, most of the dreams that we remember is coming from during REM sleep. Now, what do we mean by that? One of the things that when we talk about dream, what we remember is what the visual description or hearing description. In that sense, what is the difference than so-called dream or psychiatric symptoms? So there is an involvement of cognitive things and psychiatric inputs. We do not know all these very clearly, but therefore I was bringing in this slide, but I have not brought in all those slides, but recent papers in neuropharmacology. See, this is a model where only why REM sleep is coming or not, the mechanics. Now, to answer your question, which was my next step, when we talk about so-called vividity of the description of REM sleep, there is a role of your amygdala or such your limbic cortical inputs. Therefore, we talked about amygdala, we studied amygdala, and we talked about VTA, ventral tegmental area, and we are talking about other central nuclear there. What it seems that they are feeding to these REM on neurons and REM off neurons, and in the process, they are triggering somewhere the dreams are coming. Now, what is the meaning of somewhere what I am saying? The major problem to study these experimentally, if for dream what you are referring to, if I dream something, unless I tell you, you will not know. There is no experimental way to know that somebody is dreaming. We can talk experimentally REM sleep. We can talk eye movement, but what is the mental activity and what is that you are dreaming? Not only that, whether I will tell you or not, even I would not know if I am insane. I have dreamt a rose, I'm talking about a cow. That is again a cognitive feeling, and therefore the internal brain circuitry has to be there. And because of that, the closest we can say that whether it is triggering those internal structure, the REM sleep related neurons. And indeed, we have studied and we are showing that our two papers, one is brain recirculating and one is neuropharmacology. And the third is under review now in neuropharmacology, which gives the closest, because in animal, we would not know whether the animal is dreaming, but that is not only in animal, human as well. So this is what is happening. So this has a relationship. Now it is happening in non-REM sleep as well. In order to do that or explain that, I have to go back to my technicality or the neuronal control. I have to take another 3 hours. I think the next speaker and others will be there, which I will not be able to take at this moment. And your second question was, the question is why during sleep? That is because in order to do that, the brain wave has to come down. Because the energy level of the higher voltage EEG synchronized wave is not capable to do this, that remains submerged. Therefore, sometimes during awake also EEG desynchrony, you can start seeing in something. That is what you are having in case of psychiatric disorder, what we talk about daydreaming. So we can have it. But then in order to have this thing, you have to have the brain wave has to, that is my hypothesis. I do not have any evidence because it will be difficult to prove that way. And this is what is happening in this sense. What's your question?
So second question is there any relation between the gut microbiome and
gut
REM sleep?
I understand. Yes. I would not be able to tell you any direct evidence for it, but as you can understand, if something is being regulated by the brain, the gut, when something is being fed, if it is absorbed and you are absorbing any molecule, so be it gut microbiome or any molecule which might affect at the brain those neurons, be it any neurotransmitter level, that is likely to get affected depending on what level and other things. So I am sure they will be affecting, but then that will be affecting anything. Sometimes like you are getting affected if you have walked 100 miles or you are very tired. Sometimes it can be affected without doing any work if you are not doing any work and therefore you are depressed. So depending on that, depending on the level of neuronal environment, it will be affected. Sure, it will be affected, but and you will get anything, whatever you do, something will be there. Yes.
Thank you, sir, for your nice lecture. Today we have learned that noradrenaline can act as a double-edged sword in our brain. Thank you again, sir. We are really enriched with your lectures. Hope you will again come again to our college to enrich our knowledge about the higher brain functions, and particularly in the REM sleep, in the field of REM sleep. Thank you, sir, and regards to you.
Any other question or anything from anybody?
Any questions? Or we proceed with the second part?
Good morning, sir. Hello.
Good morning. Yes, good morning.
I have a little doubt about it. It's a very nice lecture, and we really it is nice to hear you. And sir, I was thinking like whether it is possible that the noradrenaline, the impact of the sleep on these neuronal cells, can it be slipped into other systems also?
What it is echoing? It's what happened in other system?
Like is it very specific only to those brain neurons or any other neurons or any other system?
No, for example, if you see the slide, let me see. Can you see the slide now?
It's not there, sir.
Okay, I just stopped. Let me see. I will just share. Hold on. Yes, so now, can you see the slides?
Yeah, we can see.
Yes, can you see this slide now?
Yes.
See, this is locus coeruleus. If you see my pointer, this is locus coeruleus neuron. That is a central place in the brain. And if you see, that gets input from be it preoptic area, which is known as thermoregulation, dorsal raphe, 5-hydroxytryptamine, which is important for emotion. These are the known things. It may do many other things. Like these areas, then hippocampus, substantia nigra, amygdala. So when these are there, they are giving input and they can modulate these neurons. And this will release noradrenaline, and that can affect the brain as well, which you can see in the right panel. Look here, noradrenaline is a central place. We are talking about environment. We are talking about at the cell level and at the gene level, and we are talking about the whole behavior. And this noradrenaline can affect the neurological function, the cardiac function, the mental function, the immune function. These are memory function. These are a few. Means this noradrenaline is projected all over the brain. And if it is there, if it can modulate that particular part of the neurons, which is suppose modulating heart function or respiratory function or doing any other function that way, it will affect. And then through that, it might go, suppose, at the endocrine level. So that hormonal changes will take place. In turn, the glucose will get affected, and therefore you are getting hyperglycemic. then it will be affecting the thyroid. And therefore, if there is a sleep disorder, any or all these functions are getting affected depending on how much regulation that part is taking place or not, depending on predisposition of that part. Therefore, in most of these situations, REM sleep is getting affected. Or if REM sleep you are trying to maintain, I would even extrapolate something is happening by meditation and things are getting affected, which we do not have the understanding as yet. I would love to do all those things at these stages, provided I get time and other things. But yes, it will get affected, certainly, no doubt.
So it is like something like a vicious cycle that once it is started and it may go into a vicious cycle, so that it is worsening the situation until unless it is going to be stopped by some other means.
See that with that way, if something is so fundamental, if you talk about glucose, everywhere brain needs so much of glucose. If you get affected and you say that everything is affected, it certainly will be having. For example, if I meet with an accident with her by just clashing with another person, I might get little bruise or something. I might get accident with a bicycle, I might recover within a day or two. But if a truck smashes on me, if it goes through the leg or my hand, the whole thing might get damaged. It depends on the intensity and global effect, the integrative level, what is happening. So it will be very difficult to comment right now. For that, probably one can talk about what we call these days as synthetic biology at the cell level. How much is enough? What is the threshold for every level? That would also depend on the background and then to go through. But then if we talk about everything is controlled and doing only one thing, then certain level one can do. But just the way you are asking, it will be extremely difficult, at least for me to comment on. It might affect globally. It might not, depends on what condition one existed.
I agree, sir. Thank you, sir. Thank you very much, sir.
Okay, thank you. So now we proceed to the second part of today's brainstorming webinar. I'm happy.
Thank you very much to everybody. Yeah.
Thank you, sir. Thank you, sir. Okay. So our second speaker is Professor Tushar Kanti Ghosh, who is a visiting professor of Department of Physiology, UCSTA. He has been working on brain and immunity for last few years. He was trained as a neuroscientist from the Center of Neuroscience, University of Calcutta, and Department of Pharmacology, Harvard University of USA. His research interests include immune regulation of brain, neurodegeneration in Alzheimer's disease, autonomic control of cerebellum, cognitive and motor development in children, and human physiology in synodic period of moon. He had received research grants from UGC, DST, NTRF, DRDO, DBT, etc. Professor Ghosh was president of the section of medical science, including physiology, Indian Science Congress Association in 2013, which was the centenary year of ISCA. So today, Professor Ghosh will deliver on cognitive impairments in Alzheimer's disease. So I warmly welcome Professor Tushar Kanti Ghosh, who is also well known as our beloved TKG sir, to deliver his second eminent lecture. So sir, please, over to you. স্যার আপনাকে আপনি আনমিউট নিজে দেখুন, ওখানে অপশন আসছে না আনমিউটের?
তোমরা কি আমাকে শুনতে পাচ্ছ?
হ্যাঁ স্যার।
ডু ইউ হিয়ার মি?
Yes sir, now
Okay then, so I would request to present my slide which I have sent you earlier. So there might be a technical difficulty, so I would request you to present my slide and I will speak from here.
Okay sir.
Okay.
Okay sir, I will just present your slide.
Hello.
Yes, sir. You are audible, sir.
Sir, I am audible?
Yeah. Yes, sir.
Yes, sir, you are audible, sir.
I don't see the slides.
Sir, your
Yes, sir.
sir, is the slide
Shatabdi is presenting your slide.
But I don't see those slides. That is a problem.
Okay.
Sir, if you have it in your computer, you please open it from there, and I am presenting so all the delegates can see the slide.
No, delegate can see the slide, but if I want to discuss, how can I discuss without seeing the slide? Okay. Shatabdi,
Yes sir.
so have you presented the slide?
Yes sir, I am on the first slide.
You are on the first slide.
Yes sir.
Okay, then what I will do, I will go to my computer to look at the slide, and then I'll be talking about my slide. Okay, let me try.
Okay sir.
Do you hear me?
Yes sir, you are audible.
Okay, so this is the first slide. So do you hear me? This is the first slide. The topic is cognitive impairment in Alzheimer's disease. Okay, do you see it?
Yes sir.
Yes,
Okay. Okay, so at the outset, I like to convey my gratitude to the organizer of this webinar from Vidyasagar's college, especially who was constantly contacting me, Dr. Shubhadeep, and also the others who are actively involved in organizing this webinar. Today, I'll be talking about the cognitive impairment of in Alzheimer's disease. So go to the second slide.
Yes sir.
In the second slide, you see the human exposed brain. The drawings are taken from the anatomy book published in 1543 by André Vesalius from Belgium. The name of the book is On the Fabric of Human Anatomy, and the drawings are made by great painter at the time, Titian. This is ink drawing. So this is one of the oldest picture of the brain, human brain, and you can see in the right side of the picture, you can see the corpus callosum in between the two hemispheres. So the human brain attracts the scientists for a long time. And next slide. Next slide, please.
Yes sir, I have done.
Okay. In the next slide, you see a word. The meaning of this word is brain. This was written in a papyrus, Egyptian papyrus, 7th century BC. So 7th century BC, in 7th century BC, the people of Egypt, they tried to write brain in their letter at that time. So this is the first evidence of the brain written on papyrus. Next slide, please.
Yes, sir.
So this brain attracts human from very ancient time because of its importance. Importance means its functions. So these functions are well known. The sensory function by which we can sense, and the motor function by which I can talk or you can walk. But the higher functions of brain, which is the focal theme of this webinar, that is most important. So what are the higher functions of this brain? These are learning, memory, attention, language, emotion, decision making, and problem solving. Also the consciousness is a higher functions of the brain. And in the last lecture, you are listening to this aspect of the brain, how this consciousness is in fact changed in a living brain in different state, wake, non-REM sleep and sleep. And the human brain as it is huge one, you can see next slide please.
Yes, sir.
Sensory, motor and association areas. In this slide, you can see the human brain, and we know the red area, that is the motor area, the central gyrus you can see in the middle, and the greenish part, that is the sensory area, and also the visual area. But what are the other functions? You know, these large areas, they are called association areas. These association areas of the brain, prefrontal cortex, that is parieto-occipital cortex, temporal areas. So these large areas of the brain, they are in fact related with the higher functions of brain. Next slide, please. Okay,
Yes, sir.
cognitive functions. So in the cognitive functions, which I want to discuss here, the cognitive function, what do you mean by cognitive function? Brain cognitive functions are the mental processes that allow us to receive, select, store, transform, develop, and recover information that we have received from the external stimuli. This process allows us to understand and to relate to the world more effectively. So in the cognitive function, if you look at all the aspects of the cognitive function, most of the higher functions of the brain are responsible. That means there is attention, there is memory, retrieval of the memory, problem solving. Most of the higher functions of the brain are related with this cognitive function. Next slide, please. Next slide. Neurological disease are windows in the wall. Is it visible? Hello?
Yes, sir. Sir,
Yes, sir.
You continue. Yes.
Okay, neurological disease are windows in the wall. Now, for the motor and sensory function, the functions may be deciphered by experimental means. Of course, disease can also help them. But for most of the cognitive function in human brain, it is difficult to understand the mechanism of those functions because these large association areas, they are highly complicated, they are interconnected with each other, and the functional operation of these areas are difficult to be deciphered in experimental condition. But nature has made its experiment, so there are neurological diseases. These neurological diseases are just like windows in the wall. So as you see, the sky and the outer world through the window. Similarly, through those neurological diseases, neurologists and neuroscientists are able to get the glimpse of the mechanism of the neurological disease. Next slide, please.
Yes.
Dementia affects many cognitive functions. Dementia is a disease. So what does it mean? The common symptoms of dementia is memory loss, difficulty in concentrating, finding it hard to carry out familiar daily tasks, struggling to follow a conversation or find the right word, being confused about the time and place, and mood changes. So dementia, in fact, is disorder of many cognitive functions. And dementia affects many people. So this is one neurological disease. Next slide, please.
Yes, sir.
Alzheimer's disease is a type of dementia. So today I'll be talking about Alzheimer's disease, cognitive impairment in Alzheimer's disease. Alzheimer's disease is in fact a type of dementia. So in this disease, lots of cognitive functions are affected, like amnesia that affects memory, aphasia, language, there is anxiety, poor judgment, emotional change, and lots of other cognitive changes occur. Next slide, please. So in my lecture, so this is the major points. In my lecture, I'll be talking about the symptoms of AD patients, pathophysiology of Alzheimer's disease, molecular neurobiology of Alzheimer's disease, experimental animal models to study Alzheimer's disease, and neuroinflammation as a cause of neurodegeneration in AD. In fact, in my lecture, I will try to explain the mechanism of neurodegeneration, because if we want to develop drugs, then the mechanism of neurodegeneration is very important. So why the neurodegeneration occurs? If we are able to understand the mechanism of neurodegeneration, then the suitable drug may be developed. In fact, there is no drug which can cure Alzheimer's disease. The drugs are there which can slow down the process or can improve the lifestyle, but there is no curable drug. So in my lecture, I will try to point out this point. Next slide, please. Symptoms of Alzheimer's disease. As I mentioned earlier, the Alzheimer's disease affects many cognitive functions, including memory. Initially, the recent memory or early phase of the memory are lost, not the remote memory or past memory. The early memories are affected initially, but as the time goes on, the other memories are also affected. Difficulty in identifying the meaning of simple words or numbers. So the person, you see, it is another cognitive function. The person is not able to understand the meaning of word or number. There might be general confusion, agitation, delusion, social disinhibition, and paranoia. The exaggerated personal disorder. Language disabilities is there, often mute. Densely amnesia for both recent and more remote events later on. Next slide, please. So, AD is a neurodegenerative disease. What do you mean by neurodegenerative disease? If you look at the brain of the upper one, left one, that is normal brain. But other two brains, the lower one and on the right side, they are AD brain. In the AD brain, you see the sulci are extended. The gyrus, as you see over here, it is not like normal one. The brain weight is decreasing. It is shrinking. Now, why it is shrinking? Because, brain is made up of neurons. and also neuroglial cells. In AD, these neurons are degenerated. So whenever it is degenerated, there are anatomical abnormality in the brain which can be shown from this gross brain structure. Next slide, please. The first report in 1906, Alzheimer's disease is named after Alois Alzheimer, a German scientist, a neurologist in fact, reported a case study of woman in her late 40s who developed memory impairment and deficit in other mental faculties that worsened in severity until her death in her early 50s. So this was the first report, but at the time it was a kind of dementia. But later on, this type of dementia, well characterized by, reported by Alzheimer, is named after him. So it is called Alzheimer's disease. Today, about 26 million people worldwide suffering from Alzheimer's disease. Demographic trends indicate the patients of Alzheimer's disease will increase to 4 times by 2050. Next slide, please. AD deaths in different countries. Do you see this slide? So different countries of the world are shown over here by the color, the deep red color. Deep red color indicate about 12 deaths per million. And the yellowish one, data are not obtained from those countries. So if you look at these countries, whole North America, Canada, USA, and also the European countries, West European countries, West Africa, and also part of Asia, Australia, and also the South America, the Brazil, in those areas, the huge number of human beings are affected by this Alzheimer's disease. So this is a worldwide problem and we know very little about this disease. Next slide, please. Progressive neurodegeneration in AD with symptoms. So even if there is no symptoms, the disease may set in at least 10 to 20 years before the symptoms appear. The disease may set in. That means the nerve cells are being degenerated. And whenever the cells are degenerated, the functional derangement may not be found. The functional derangement means initially the memory loss is the first sign of AD, because as you see in the left lower figure of the sagittal sections of brain, do you see this blue color? This is entorhinal area. The neurodegeneration occurs initially at the entorhinal region of the brain. And these regions are related with hippocampus. This entorhinal hippocampus pathway, they are related with the memory consolidation. So, whenever there is neurodegeneration, memory is not initially affected because there are huge number of neurons. Nature has given us lots of neurons, and in fact neurons are dying even after birth. The neurons are decreasing in number in the brain from birth, but functional abnormalities are not found because of neural connections are formed by the existing neurons. But when the number of neurons are decreased more than 40%, 50%, then the functional abnormalities appear. As the region is related with the memory consolidation, so the initial change is in the memory, but in the preclinical condition, this neurodegeneration has started, but there is no symptom. So whenever you see the symptom, large number of neurons are in fact degenerated, and that's why you see the symptom. And the problem is, whenever there is severe loss of neuron, it's very difficult to repair because neurons hardly regenerate in the brain, except in some regions these neurons are not regenerated. So functional recovery is a problem. Next slide. Progressive neurodegeneration in AD with symptoms. In mild and moderate AD, if you look at the left coronal section of the brain, then you will see from the preclinical side, the cerebral ventricles, they are enlarged a little bit, and the cortical shrinkage is also prominent because the sulci are getting widened. If you look at the lower sagittal cartoon, then you see the neurodegeneration is not limited in the entorhinal area. It has spread to the temporal region and also the prefrontal region, other association areas. That means they will affect other cognitive function. So initially it may be the recent memory, but with the time, whenever the more and more neurons are dying, so there will be other memory problem, other cognitive problem, which include, as I told you, confusion, trouble in handling money, poor judgment, mood change, increased anxiety. And next slide, please. Whenever there will be severe AD means, so whenever large areas, as you see in the lower diagram, the blue color, they have gone to the temporo-occipital region, prefrontal region, temporal area, and in the cingulate gyrus. So they are in fact widespread. And due to the death of the neuron, look at the distended ventricles, lateral ventricles in the brain coronal section. So here, the other abnormalities will come, that is the weight loss, seizure, skin infection, groaning, moaning, gurgling, increased sleeping, loss of bladder and bowel control. Death usually occurs from aspiration pneumonia or other infection. These persons are very susceptible to infection because their immunity is probably compromised. Next slide. So whenever I am talking about these association areas, entorhinal cortex, hippocampus, cingulate gyrus, I want to mention to the students about the neural circuitry within these regions. They are median cerebral bundle, dorsal tegmental fasciculus, weak the other tract, which connect, as you see over here, which connect the mammillary body with the anterior nucleus of thalamus. So all these fibers are very important. Most important is the connections between entorhinal cortex and hippocampus. In fact, lots of association areas, they are connected to hippocampus. Next slide, please. As you see over here, the unimodal and polymodal association areas, that is frontal, temporal, parietal cortex, they are connected to parahippocampal cortex. If you look at the circuitry at the lower part of the right hand side, they are parahippocampal and perirhinal areas, they are connected with the entorhinal cortex, and from entorhinal cortex the informations are going to dentate gyrus by perforant pathway, and from dentate gyrus to hippocampus CA3 region by mossy fiber, and then Schaeffer fiber, through Schaeffer fiber hippocampus CA1, then subiculum coming back to entorhinal area. So this neural circuitry is very important for consolidation of memory. Now, whenever there is degeneration in the entorhinal region, as I told you, and hippocampus, obviously the memory will be affected first. Next slide, please. So the memory is affected, but in other diseases also memory is affected. Memory may be affected. Neurological disease, there might be different types of dementia, but the neuropathological hallmarks of Alzheimer's disease is plaque and neurofibrillar tangle. So you can identify Alzheimer's disease from the pathophysiology of the brain. In the brain of Alzheimer's patient, there must be plaque and neurofibrillary tangle. In other dementia, neurodegeneration may occur. There might be memory loss, but these neurohistological or pathological characters are absent. They are only present in Alzheimer's disease. So what is plaque? Plaque is extracellular deposit of amyloid beta protein which are surrounded by a cell of dystrophic neurites. The neurotic component of the plaque consists of morphological abnormal dendrites and actions of different types of neurons, typically associated with substantial number of glial cells. So there is a core of plaque which contains amyloid protein. These amyloid protein, there are different species of amyloid protein. I'll be talking about that. But they are covered with a shell of neurites. So they are the processes of neurons, and also there are glial cells. So these plaques is histological characteristics of Alzheimer's disease. And there is neurofibrillary triangle. Within the neuron, there are intracellular densely packed abnormal fibers that occupy much of the cytoplasm and proximal neural process. These fibers in many tangles bearing neurons are so high in number that the nucleus is displaced and the cell body appears to be swollen. So these fibers, they are not present in the normal neuron, but whenever these fibers are present, they are filling the cytoplasm. So these they are filling the cell body and the actions, and by that way normal functions of this neuron, that is exoplasmic transport of material, that is not possible. So that is affecting the neuronal activity. Next slide, please. Here you see the histological picture of plaque on the left side and the tangle on right side from the human brain. In the left side you see those plaques with core beta amyloid protein, and in the tangles you see the fibers within the cell. Next slide, please. So in the core of the plaque, they contains A beta 42 amino acid containing amyloid beta protein, or 40 amino acid containing amyloid protein. But today scientists are reporting that there might be other species, 25, 30, 40 different species of A beta 42 in the core. Next slide, please. Biochemistry of neurofibrillary tangles. The tangles consist of pairs of twisted filament with a highly regular periodicity. They are called paired helical filament. So PHF, they are composed of cytoskeletal protein, the microtubule associated protein tau. So microtubules are normal component of the nerve cell. They are stabilized by tau protein. Now in Alzheimer's disease, these tau proteins are hyperphosphorylated. They get dissociated from the microtubules. So microtubules are in fact disintegrates, and these tau proteins form this paired helical element. Next slide, please. So in this cartoon, it is nothing new. Though there are billions of neurons in the brain, they are interconnected. So they communicate with each other, carry out metabolism and repair themselves. Now this communication, in synaptic communication, that is affected, initially affected. Whenever there is no neurodegeneration, the synaptic connections may be affected initially. And then there will be neurodegeneration. So AD disrupt all three of these essential functions, that is communication, neuronal metabolism and repairing. Next slide, please. In this slide, in this cartoon, in fact, you see the tau protein which are stabilizing the microtubules. They are dissociated. The microtubules are disintegrated, and these tau proteins, they are forming those paired helical strands in the which are the component of the tangles. So the microtubules are not able to stabilize the neuron. So this is the pathology. Next slide. the American Association of Aging Alzheimer's Disease, NIAAA, they said parameters for identification. How will identify the AD disease? There should be amyloid plaque density. So in the brain, PET scan can identify the amyloid plaque in the brain. So amyloid plaque density may be assessed whenever there is higher amyloid plaque density, that is indicator. Brain metabolism assessed by 18F-FDG in PET. So neurons are utilizing glucose. So whenever there is problem in utilizing glucose, that can be identified by 8F-G PET. And hippocampal volume by MRI. As the hippocampus is affected, the volume is decreasing. So these characters or parameters are used for identifying a patient whether he is suffering from Alzheimer's disease. Next one. The changing brain in Alzheimer's disease, here you see in the left one, this is normal brain. And right one, this is PET of Alzheimer's brain. Here you see the oxygen is not supplied to many of the association areas of the brain because the neurons are not active, they are not normal. So the utilization is less, and that is one of the feature to identify AD. Next one. Terminology of classification. National Institute of Aging, Alzheimer's disease, they have classified Alzheimer's disease into different groups. In the preclinical stage, as I told you, when there is no clinical symptom, there the amyloidosis, or meaning whether the amyloid plaque is accumulating over there and neurodegeneration is there. So these two characters are used to classify the stage. In the stage 0, preclinical stage, there is no amyloidosis or neurodegeneration. In preclinical stage 1, there is amyloidosis but no neurodegeneration. In stage 2 and 3, both A and N are present. In suspected non-Alzheimer's disease pathophysiology, there is no amyloid plaque, but neurodegeneration is there. So they are non-Alzheimer, but they are suffering from dementia. So these are the characters used by NIAAA. Next slide, please. Now the question is why this A beta accumulates. So there is a large protein called APP. Amyloid precursor protein is formed in the neuron, and these molecules is processed by two pathways. One is called alpha secretase pathway and another one beta gamma secretase pathway. In alpha secretase, the component which is formed from APP are used for normal physiological function like neuronal protection or growth. But beta and gamma secretase pathway, they form A beta protein, A beta 40, 42 or other species of protein, which are formed normally in minor quantity. But in pathological condition, that means in AD, the A beta production is increased, and that is a problem. So APP processing is very important. Here, next slide please. Here you see a cartoon where you see in the left side, the APP protein are embedded in the neuronal membrane. In 2, you see the enzymes, as I told you, the beta and gamma secretase enzyme, they are breaking this large protein into 2 places, that is beta position and gamma position. As a result, in the 3rd one, you see the small segments of beta amyloid proteins are formed, and these proteins can self-aggregate. They can aggregate, and they are forming the core of this plaque. And later on, it is surrounded by other neurites and glial cell, and according to some group of scientists, these plaques are the cause of neurodegeneration. Next slide, please. However, there are 2 distinct groups of Alzheimer's disease. Only 2 to 5% of the patients are familial. That means it is related to certain genes. These genes were identified. They are APP gene, presenilin genes. Presenilin is component of this beta secretase enzyme. So these genes are mutated, and certain families are identified where this mutation is going on. So most of the members of that family are being affected by the familial AD, but only 2 to 5% of the patients are familial. And most of the patients, we don't know the cause of this neurodegeneration. They are called sporadic. And though the causes are not clearly known, but certain genes like ApoE genes can increase the susceptibility of this disease if the certain component of the genes are repeated. That means many copies, if they are present, especially the E4, then there is a possibility that persons will get affected later on with AD. So there are two basic groups. Initially we talked about memory loss, dementia. Then we talked about neurodegeneration. Now the cause of neurodegeneration. In familial patients, the neurodegeneration is due to the genetic defect, formation of A beta, and how this A beta is causing neurodegeneration. Next slide, please. This A beta 42 mechanism of neurotoxicity by amyloid proteins, A beta 42 appears to be responsible for neurotoxic effect. Soluble A beta oligomers impair functions of synaptic ion and glucose transporter with loss of synaptic plasticity. So whenever A beta are soluble one, they affect the synaptic activity because they affect the glucose transporter and also the ion transporter, mostly sodium potassium ion. So whenever these ions are abnormal and glucose is not able to enter inside the cell, the synaptic activity is affected. A beta 42 oligomerization are associated with microglia activation. There are lots of glial cells and the microglia are activated. There is astrocytosis that initiates inflammatory response. The inflammation is probably dependent on microglia and astrocytes. So the role of glial cells are also emphasized now for this neurodegeneration. And I told you about the cytoskeletal protein that also disrupt. So these are the mechanism of A beta 42 neurotoxicity. Next slide, please. Amyloid cascade hypothesis. In this hypothesis, this is very important one, how the familial AD sets in. APP, PS1, PS2 gene, whenever they are mutated, A beta 42 are formed in large number. These A beta 42 oligomers act on synapses and progressive synaptic and neuritic injury occurs. Microglia and astrocytic activation also occur. They secrete cytokine and other neurotoxic material like ROS, nitric oxide, and many other reactive oxygen species. So they can destroy the neurons. Altered neuronal ionic homeostasis results in oxidative injury, and they also altered kinase phosphatase activities that result in the formation of tangles. So tangles are formed, and from the plaques, all those inflammatory materials are released which are neurotoxic, and they cause neuronal death, which is the cause of dementia. So this is the cascade hypothesis. This is fine. This has been proved in experimental model of rat. So there are genetic model of AD, and scientists have proved this pathway very nicely. So there is no problem. But the problem is only 2 to 5% patients are from familial one. So what happens to the other? Now there are problems in the next slide, please. Problem in central amyloid hypothesis. Progressive dementia cannot be correlated with plaque number. Now, many neurologists, they are reporting that the number of plaques are not increasing with increased dementia. So if the plaques are cause of neurodegeneration, why their number are not increasing? suspected AD pathology lacking A beta deposition but with neuronal injury are converted to mild cognitive impairment in 10% cases within 1 year. So this is another problem. But most important problem is the neural injury can at least partly occur independent of A beta related process. In many patients, the neuronal injury may occur independent of A beta. That means A beta may be a cause of neurodegeneration, but it is not the only cause. And the drugs developed on the basis of amyloid hypothesis. So the drugs which will inhibit the accumulations of A beta, or they will affect the beta secretase pathway, gamma secretase pathway. Lots of drugs have been developed by the pharmaceutical industry, but these drugs are not effective in all the patients. Next slide, please. Is AD multiparametric pathology? That means AD is a disease with single sequential pathological pathway, with A beta considered as the only initial causal event, is likely to be progressively replaced by more complex picture, in which AD is considered as a multi-parametric pathway, that is subtended by several pathways independent of pathologic process. So gradually, considering this sporadic AD, it becomes clear that A beta is not the only problem. There are other causes. Next slide, please. So to understand this mechanism, the sporadic mechanism, the scientists are trying to develop the sporadic animal model by different ways. It is not the genetic model. It is sporadic model. Usually those are prepared by injecting certain material within the brain. And in my laboratory, we have developed many animal models of sporadic AD. These animal models are used to investigate the pathophysiology of this disease, how this sporadic disease is initiated. And in the next part of my lecture, maybe I have some time, I guess, maybe 10, 15, how? 10 minutes. 10 minutes. Okay. 10, 15 minutes. Okay, I'll be covering the result. So next slide, please. So here you see how these animals are prepared stereotaxically. So by drilling the brain and by injecting colchicine, which can dissociate those microtubules. It is plant alkaloid, in fact, or streptozotocin that can also be injected into the brain, or A beta can also be injected. So there are ocadaic acid may also be injected. There are lots of chemicals which can be injected in the brain in measured quantity into the lateral ventricles on the both sides. And after some time, these animals will show the problems of cognition. Their memory is affected. That can be assessed by different techniques like radial arm maze, or their histopathology may be identified by staining those plaques or the nasal granules, which indicate the functional status of the neuron. As well as we can probe into the neuroinflammatory markers into the brain. So in the few slides, I'll be talking about those results. So colchicine, a plant alkaloid, was injected into the lateral ventricle, and the memory was identified, working memory, reference memory, and other sorts of memory. I'll be talking about two because it's not for use discussing all the memories. Neurodegeneration, the histological, histopathological character of neurodegeneration, comatolitis or plaques, and inflammatory markers in the hippocampus. So here you see the radial arm maze data on working memory. Working memory means there are bedded arms and non-bedded arms in the radial arm maze. So whenever the rat has entered into bedded arms and wrongfully it is going again to the bedded arm, then it is working memory error. So here you see before the injection, ICV injection, as you see in this graph, so day 5 it was injected. Before that, there is no error. But after injection, if you look at the upper one, the error is gradually increasing with time. This is 21-day study, and the error is gradually increasing. That means the cognitive functions is affected in this brain. Now, in those rats, the neurodegeneration was tried to, certain drugs were injected to inhibit this neurodegeneration. Colchicine usually increases COX-2 enzyme activity. So there are certain drugs, non-steroid anti-inflammatory drug, heterocoxib, which inhibit COX-2, that was given. And you look at the reference memory error in those treated animals at the lower part, their errors are decreasing. That means colchicine can initiate the increase the cognitive impairment. The working memory may be affected, error is increased, but whenever we are treating those animals with heterocoxib, a COX inhibitor in graded dose, the different 20, 10, 20, 30 milligram per kg, the error is gradually decreasing. Okay. Next one. The reference memory error, I don't want to spend time over here. It's the same thing, only the reference memory is totally different one. It is whenever the animal is entering into non-baited arm, the animal is not trained to enter in those arms. Normal animal will not enter over there, but whenever there is problem, they will not be able to identify baited arms and non-baited arms. So they will enter into the non-baited arms, and this is reference memory error. And here you also see the similar trends. Now, if you look at the crystal violet staining of the control in the left side, control SAM operated. SAM operated means here certain artificial CSF was injected into brain, and AD animal where colchicine was injected. If you look at the hippocampal nasal granules, you see in the AD animal the C1 is 15-day study. From 15-day study, C2 is from 21-day study. So gradually the nasal granules are decreasing, indicating the functional status of the neurons over there. So they are affected. And if you look at the right-hand panel, 15 days and 21 days, whenever heterocoxib was given, 20, 10, 20 and 30 milligram, if you look at the 30 milligram, nasal granules reappear, suggesting the colchicine-induced neurodegeneration, rather the new chromatolysis may be inhibited by these drugs. Next slide, please. Okay, don't want to discuss. Next slide. So Congo red staining of amyloid plaque. Congo red, as you see in the left one, in the control there is no plaque. Some no plaque, but in AD you see those. You see the arrow; those are plaques. So the plaques are increasing in 21 day. Now, whenever in the right hand side, if you look at the same areas, hippocampus, whenever 30 milligram of heterocoxib was given, the plaques are disappearing, suggesting the colchicine-induced neurodegeneration. We initially found about the behavior. Now, neuropathology can be corroborated with the behavior. That means whenever there is no plaque, there is no chromatolysis, the behavior is also normal. And whenever there is plaque and chromatolysis, the behaviors are affected. Next slide, please. So I can skip that one. Next slide, please. Now the inflammatory markers are increased in hippocampus. We look at reactive oxygen species, nitrite, IL-1 beta, TNF alpha, COX-2, prostaglandin. These are measured in the hippocampal areas and also in other parts of the brain. But I am presenting hippocampus here. Next slide. So here you see the ROS level. Look at the left-hand side. This is ROS level in hippocampus. ROS level in hippocampus is increased, say 150, 200 times. If you look at the left side, in 15 days, in 2-week study, 150, in 3-week study, 200. But whenever the heterocoxis were given, they are decreasing. Same trends. Next slide, nitrite. It is indirect estimation of NO, and that is also used in the AD animal, but they are also decreased by heterocoxib. Next one, COX-2. COX-2 is one inflammatory. They cause production of prostaglandin, and that can cause further inflammation. So COX-2 was measured, and as you see over here, their concentrations are also increased, and the heterocoxib can decrease them. Same trend. Now, prostaglandin, similar one, left side. In the hippocampus, they are increased in AD animal, and compared to SAM-operated and control animals, and they are decreased by heterocoxib. Next one. So neuroinflammation, as you see, the neuroinflammatory markers are increased, and whenever they are increased, there is chromatolysis, plaque formation, and whenever there is chromatolysis and plaque formations, the behaviors are affected. So it appears that the behavior, memory behavior, especially here, neurodegeneration, they are linked. And neuroinflammatory markers are probably related. These type of studies indicate that neuroinflammation is probably the cause of neurodegeneration in this animal. They are not familial, they are sporadic, induced by certain chemicals over there. So neuroinflammation may be a cause of neurodegeneration and dementia in CAD rats, as COX-2 blocker heterocoxy was able to inhibit neurodegeneration and cognitive impairment. Next one, Shatabdi. Vitamin C. So it is 12:30. How far I can go, Shatabdi?
Sir, you have 5 minutes time
Huh?
maximum. Sir, 5 minutes maximum.
5 minutes maximum. So I will reduce my lecture. So vitamin C is antioxidant and has been used to reduce oxidative stress. So in this animal model, we have used vitamin C in different doses. And look at the result. So here, the behavior, the memory behavior, as you see here in the working memory and reference memory, they are deranged after colchicine. But whenever vitamin C at the dose of 200 and 400 are given, their behaviors are repaired. But the 600 at 600 mg, their behaviors are not repaired. In fact, it is in fact exaggerated. So vitamin C is showing two opposite results: lower concentration, they are reducing neuroinflammation probably, and higher concentration, they are probably affecting the impairment, just like the colchicine animal. So that occurs in both the behavior. And if you look at those chromatolysis, if you look at the lower one, next slide please. Next slide please. If you look at the chromatolysis, if you look at the lower one, you see 200 and 400, they are inhibiting chromatolysis, but 600, that is increasing. Next slide please. So plaque, if you look at the plaque, 200 and 400, they're decreasing the plaque. At 400 and 600, it is increasing. Next slide, please. So if you look at the inflammatory marker, they are also showing biphasic response, 200 and 400. Here you see IL-1 beta, TNF alpha, they are lowered in 200, 400 and increased in 600. Next slide, please. So here the ROS level and nitrite level, here the same trend is found. Next one. Next slide, please. So summary of vitamin C study. Vitamin C at lower dose in CAD rat reduce memory impairment, prevent neurodegeneration, inhibited neuroinflammation. But higher dose, that is the opposite thing. Dual role of vitamin C in colchicine has been proposed from our study. So that has been reported. So this has been accepted. And we have explained why it can be pro-inflammatory or anti-inflammatory in different doses. We don't have time. So next slide. Next slide. Dual role of vitamin C. So next slide. After dual role, inos may play an important role in neurodegeneration. So long we are talking about vitamin C, but in inducible nitric oxide synthase, we also tried to understand the mechanism of this enzyme by using, next slide please, by aminoguanidine that can specifically inhibit inos. Next slide please. So aminoguanidine was given in 30 to 50 milligram dose, and those parameters were again measured. Next slide please. And if you look at those, next slide, if you look at the reference memory and working memory, they are in fact, the error are reduced by 30 and 50 milligram of aminoguanidine. So they are effect, but they may not be as effective as heterocoxib, but they are partially probably reducing the oxidative disease. Next slide, please. In the next slide, as you see, the aminoguanidine is also decreasing the number of plaque. Next slide, please. The left side, the ROS and nitrite levels are also reduced by aminoguanidine. Next slide, please. TNF alpha, IL-1 beta, they are also reduced in AD animal. Left side, please. And so summary of aminoguanidine study that it reduced memory impairment, prevented neurodegeneration, inhibited neuroinflammation. So the neuroinflammation, if you look at the possible mechanism, so whenever you are giving intracerebroventricular injection, the microtubules are disrupted, which blocks exoplasmic flow, increased ROS and NO generation, and increased cytokine production. So vitamin C can decrease ROS, aminoguanidine can reduce NO, but this cytokine, they can activate COX-2. That can be inhibited by heterocoxib. So activation of COX-2, that increase production of prostaglandin, that activate microglia, further production of neuroinflammatory cytokines, and they are causing neurotoxic effect. So if it is the possible pathway of neurodegeneration, then heterocoxib, vitamin C, and aminoguanidine can effectively block this neurodegeneration, inflammation, and as a result, cognitive impairment are partially prevented. Next slide, please. So NMDA receptor, I will talk only about NMDA receptor blocker, mementin. NMDA receptor is also involved in neurodegeneration in some other conditions. So we also investigated the role of NMDA receptor by using mementin. Mementin can block the NMDA receptor. So here you see the TNF alpha, left side, TNF alpha level in hippocampus. I'm not talking about serum. They are inhibited by mementin. The IL-1 beta, left side, in 21-day study, 15-day study, they are inhibiting. And also the ROS level, they are also inhibited by mementin. Next slide. Next slide, nasal granules and plaques. Nasal granules and plaques in CAD rats. So here you see also the plaques are decreased by mementin, and the nasal granules chromatolysis are also inhibited. Next one. So mementin can inhibit neuroinflammation and neurodegeneration in hippocampus of colchicine-induced AD rat. Next slide, please. So mechanism of this neurodegeneration in colchicine-induced AD rats, whatever we got information from this experimental study, it can be said that production of ROS, membrane lipid peroxidation, and generation of 4-HNE, probably they are increased in neuroinflammation, and nitric oxide increase oxidative and nitrosative stress and mitochondrial injury. Neurotoxic cytokines are released by inflammatory process. And also from the mementin study, it can be said that glutamate toxicity is also involved. Release of glutamate from glial cell may cause excitotoxicity. And A-beta accumulation on NMDA glutamate receptor results in increased intracellular calcium, which activate cascades involving neuronal of nitric oxide synthase. So glutamate NMDA receptor blocker study also indicate that inflammation may be induced by COX, but also by glutamate, the neurotransmitter, which can affect the glial cells. Next slide, please. So cognitive impairment, so far I discussed, that is about neuroinflammation by different ways, but there are also other hypotheses which I didn't consider, that is cholinergic hypothesis, and other component like poor functioning of blood-brain barrier, disrupted homeostasis of biometals such as copper, iron, zinc, air pollution, gum disease that can also initiate AD, retrogenesis, smoking. So these are the components which can initiate neurodegeneration, but how they are causing it, whether it is the inflammatory process or other mechanism, it is not very clear. Next slide, please. So genetic and environmental factors are involved with the neurodegeneration in AD. And in this cartoon, you see the genetic factor, they can increase A-beta pathology, tau pathology, but there are other factors like vascular lesion, neuroinflammation, connectivity, abnormalities, etc. So this is a composite one which indicate that the AD is multi-parametric. So we know very little about the neurodegeneration mechanism, and from our study, we can indicate that in sporadic one, the neuroinflammation may be a major cause. Next one. So conclusion is cognitive impairment in AD is due to neurodegeneration. Neuroinflammation is a cause of neurodegeneration. Neurodegeneration in AD is multi-parametric, including the participation of genetic and environmental factors. The new drug development needs deciphering of the mechanism of neurodegeneration in the AD. So it appears the neurodegeneration in different patients are not similar, and therefore the drug should be different depending on the types of patients they are. Next slide, please. So this is our study supported by different granting agency, UGC, DST, National Tea Research Foundation, Government of India, and so DBT, and the research worker who worked, those Susmita, Ruksha, Pritha, Orijit, Nikki. So they have participated in this for this experiment. So that's all. Thank you. Here you see one AD patient who was a painter. So before the disease sets in, he drew his self-portrait, which is on the left-hand side, and the right one, the self-portrait at the severity of the disease. In this disease, motor functions are not affected, though the cognitive functions are affected. Motor functions are not affected. So how the person sees himself. So this is a very, very severe disease affecting the human life, but we are not able to do anything for it. But I have to draw a line. Probably I have not encroached much time. Thank you very much for your patient hearing. Thank you.
Very nice talk, Tushar. Nice to hear that again. Hear you again. Thank you.
Thank you, sir, for your very factual and informative speech. Okay, so now I request for any questions.
Sir, I am Alok Chaudhary. Shraddha.
Yes.
I have a small question. That is, in one of your slides, you have shown that Alzheimer's disease predominates in the country where alcoholic consumption is very high. Even in our country too. And we all know that in Korsakoff syndrome, where that alcoholic consumption over a period of time can lead to the deficiency of vitamin B1.
Yes.
My specific, now I have come to my question. My specific question is, do you believe that prolonged alcoholic consumption, or in other words, prolonged vitamin B1 deficiency can set up the Alzheimer's disease at earlier ages? This is the question.
Okay, thank you. Thank you. Thank you very much, Alok, for your question and your interest in the subject. Now, the thing is this, in Korsakoff syndrome, the neurodegeneration which occurred, that is in cerebellum.
Yeah.
In anterior lobe of cerebellum, and mostly that affect the motor function.
Right.
So there is intention tremor. Okay. So alcoholics, they have problem in neurodegeneration, but that is not on the association areas. That is mostly on the motor side, and motors are affected. But there is no report that alcoholics develop Alzheimer's disease. Though there is a possibility, because lifestyle is very important one, but there is no study which indicates that the alcoholics are sensitive. I mean, the prevalence of Alzheimer's disease is more on alcoholics. There is no report like this. On the other hand, it has been shown where there is environmental pollution. There is more environmental pollution. So after the Industrial Revolution in Europe, whenever the environmental pollutions are increasing, they are causing many neurodegenerative diseases, not only Alzheimer's disease, but also Parkinson's and other. There are also other neurodegenerative disease. And one of the hypotheses is this: these environmental pollutants are causing those things. Now, lifestyle is very important. Lifestyle is important means whenever you are taking antioxidative one, you are talking about vitamin D. But whenever your food contains vitamin C,
vitamin B1, sorry.
B1,
B1.
you are talking about B1, but whenever these vitamins which are antioxidative nature, like the vitamin C or other green vegetables which are consumed more, and they are balanced diet, probably moreover whenever the brain is doing little bit of exercise, whenever you go to gym and exercise, your muscles are little bit, muscle cells, the muscle fibers are enlarged. So if you work with brain, so it has been found that whenever the Alzheimer patients, they are playing cards or they are doing those chase, or they are creative, they are doing creative work, say creative writing or singing. So this creative work, whenever the synaptic mechanisms are activated by any work, so that will inhibit Alzheimer disease. So if you work your brain, if you work with your brain more, so that is not the motor function, that is not sensory function, but cognitive functions. That means memorization, attention. So if you practice more, so that will inhibit the progression of the disease. But B1, there is a possibility that B1, as you have said, that B1 may have effect. That means they can retard the inhibitory mechanism. But I have not seen any paper which are showing the effect. But there is a possibility.
But I have recently come across a paper where it is shown that the Korsakoff syndrome can accelerate the onset of Alzheimer's disease. It is only one paper.
It is
so
possible.
That's why I am interested to know whether you have enough information in your custody.
No. Korsakoff, as you have said, Korsakoff is causing neurodegeneration in other parts of the brain. And there is a possibility that if there is the person is sensitive, that means there is a preponderance, there is genetic or other factors, and alcohol can add on that. Okay, so if there is neurodegenerative changes in the person, as I told you, the disease sets in 10-20 years before the clinical symptom. So whenever the disease started long time back, at that time, certain oxidative stress component, they can facilitate the disease. And in this regard, the alcohol, vitamin B1, they may also participate there.
Okay, thank you, sir. And regards to you, sir, thank you.
Okay, all right.
Any other question you have for Professor Ghosh?
No question. I have.
Okay.
Sir,
Yes.
hello, sir.
Hello.
Sir, I have a little doubt. In echoing with Dr. Ghosh,
what is your name? What is your good name?
I am Prasunpriya Nayak, sir.
Where from?
AIMS Jodhpur.
Okay, Prasun, I can understand you. You can ask question.
Yeah, please, sir. Thank you, sir. Sir, echoing with Dr. Ghosh, that alcohol, what you have also suggested, the same thing, that alcohol may increase the chance of oxidative stress in many ways. And along with that, if there is something like ecotoxicity, glutamate exotoxicity could also be increased.
Right.
Sir, I have seen in my experiments that even if the oxidative stress has been inhibited, still there is some chance that behavioral toxicity, neurodegeneration is not stopped. I have done this with vitamin E. So, those amyloid hypothesis include the oxidative stress, as a part of its AD generation, AD pathology. But my idea, my suggestion is like I have suggested this that it may not be fully working through that only pathway. It, as you have mentioned, it might be multifaceted pathway.
Right, right.
However, sir, I had a question. Like as I was following your lecture, I surprised that I was surprised that you mentioned aluminum toxicity. But
So what is your question? Will you please repeat it? I don't hear it.
sir, what about aluminum toxicity?
Oh, okay. Aluminum toxicity. Is there any other question?
No, sir. I just wanted to know that you didn't include even in the environmental pollution or in the mechanism, nowhere you have included the aluminum toxicity.
Okay.
Is there any
Thank
specific
you.
reason?
Thank you, Prashut, for mentioning that thing. I didn't mention many things. I did not mentioning many things because of constraint of time. As you know, but not only those ocadoic acid, streptozotocin, A-beta-42 are used for preparing animal model, but there are also other ways. As you said, aluminium is also used because they are also causing that neurotoxicity. And not only aluminium. There are pesticides. There are also pesticides model. These pesticides, aluminum, and there are also other material, glutathione. There are lots of other material which are used by different scientists to produce neurodegeneration and specifically AD animals, sporadic AD animals. So literature is there. I mention only few, but there are also other like aluminium. As you see, there is aluminium model is there, but aluminium model is nowadays they are not using. It was little older model because it is not specifically affecting the hippocampal area. It is affecting large part of the brain. Okay, pesticides, they are also used for preparing these animal model of neurodegeneration, but that may cause also Parkinson's disease. Parkinson's models are also used by pesticides. So specificity of AD animal model, that is gradually coming to certain neurochemicals which can affect only the neural circuitry passing through hippocampus. But you are right, aluminum, pesticides, and also other drugs, they are also used for this purpose.
Thank you, sir.
Okay. Thank you. So I think we have finally come to the end of today's event. It was very factual and educative talk by both our eminent speakers today. So no program can be conducted successfully without productive and prolific participation of each member who are associated with the organizing committee as well as organizing today's national webinar. So right now, I feel delighted to mention that our Honorable Head of the Department, Dr. Subodip Ganguly, who very efficiently and competently supervised the whole event. So I request our HOD to give the vote of thanks and congratulate both the eminent speakers who have given a very illuminating and amazing talk.
Thank you, Shatabdi. On behalf of the Department of Physiology, Vidyasagar College, I deem it a great honor to propose the vote of thanks for all who have helped us in making this webinar such a resounding success. First of all, I want to thank our eminent speakers, Dr. Birendranath Mollik, Professor and Director, Amity Institute of Neuropsychology and Neurosciences, and Professor Dr. Tushar Kanti Ghosh, Visiting Professor, Department of Physiology, University of Calcutta, for adorning their outstanding scientific contributions in the field of neurophysiology. One of the most complicated area in the field of physiology, where most of the students, even some faculty members, are also having difficulties in this area. So we are really grateful to both of them for showering the audience through their phenomenal lectures. Though these topics are generally considered as difficult for students and even for many of the faculty members. But I sincerely believe that through this luminous webinar, our audience can able to find out a way to rethink about this emerging area of neurophysiology. Thanks a lot, sir, for your thought-provoking golden speeches. I would like to propose hearty vote of thanks to our honorable principal, sir, for gracing today's webinar. I want to thank our IQAC coordinator, Dr. Alaghush Choudhury, organizing committee members, Dr. Chandana Sarkar Banerjee, Dr. Oindrila Chattopadhyay, Dr. Sanatan Mistro, Dr. Monalisa De, and Miss Shatabdi Ghosh, volunteers, Ankush Kumar Das, Suman Parui, and Soham Chattopadhyay for working hard for the past few days to make the program successful. I cannot thank everyone enough for their involvement and willingness to take out on the completion of the tasks beyond their comfort zone. Our special thanks goes to Professor Dr. Debashish Bandyopadhyay, Senior Most Professor, Department of Physiology, University of Calcutta, and Undergraduate Board of Studies Chairman. Despite of his very busy schedule, he has already spent his valuable time for making this webinar successful. Many other academic personnel I have just found from the participant list, like Professor Dr. Prasuntiya Naik, Professor of Physiology, AIMS Jodhpur, Dr. Sandeep Sinha, Department of Physiology, University of Vidyasagar University, Dr. Bornali Roy Basu, Department of Physiology, Surendranath College, Dr. Devolina Ball, HOD, Department of Physiology, Shuri Vidyasagar College, and many others. They have also joined here. Thanks a lot for their valuable joining. Finally, the honorable delegates, our most beloved students who have turned up in such great numbers, not only from our college, but also from other institutions of the country. Thank you so much for your active participation. Once again, I thank all for your cordial cooperation. Thank you. Thanks again.
Thanks a lot to everybody. Bye.
Okay, thank you all for attending today's one-day national webinar. Please fill the feedback form to receive your e-certificate. The form will be active till 5 p.m. today. So see you in our next webinar. Goodbye. Thank you.
Thank you.
Thank you, sir.
Thank you very much. Alok and others. Thank you.
Thank you, sir.
Thank you, sir.
you.